Well, good morning, everybody. Hear me okay? So, I was asked to come up with defining what we call an ideal, how do we define an ideal feed additive? What is that? And furthermore, how do we assess that? And we've heard a lot of talks this week about different feed additives, what they do from a functional standpoint, that sort of thing. And one of the things that I think we have to keep in mind is that, what kind of criteria are we going to use here? Obviously, there's several different factors we need to consider. And so we have precedent for this, interestingly enough, is if we go back to antibiotics, I came across this bulletin. I was cleaning out a former professor's office at one time. And there was this book from Pfizer, that pamphlet was put out in the 1950s, and it actually is the blueprint for what an ideal food additive is. They were obviously selling terramycin, and it was all about terramycin applications for food animals. And so they went through growth performance, gut health, and a number of other things that we've been talking about all week. And so I could probably stop right there and we could just read this pamphlet and it would tell us what we would need to know in terms of, what an ideal feed additive looks like. Of course, we all know what happened is with antibiotics is at the time, ideal was easy to use, cost-effective, consistent. And that was the point that they were making in the pamphlet over and over again. We know since then, obviously, that antibiotic resistance has emerged in human clinical disease, has continued to increase and climb. And so now we worry about MAR and multiple antibiotic-resistant bacterial pathogens. That's led to public perception, and certainly in the U.S. that's been a driver is public perceptions. We don't want antibiotic-fed animals. We don't want antibiotic-resistant pathogens, etc. And so what we've seen now is a complete push from a market standpoint to take antibiotics out of pretty much all food animal systems. The McDonald's of the world, the fast foods of the world have basically told their suppliers, we want to market meat products, fast food products, etc., that come from animals that have not been fed antibiotics. And so, at least in the U.S. anyway, antibiotics are pretty much gone. So, what's happened in conjunction with that is we've seen a big jump in livestock production without antibiotics. And so, of course, we have antibiotic-free from a conventional standpoint. The large organizations such as the Tysons of the world are certainly marketing antibiotic-free type chicken products, etc., pork products. The rise of natural raised products, pasture flock, free-range poultry has become more and more of a thing in terms of a market. Of course, the organic production in the U.S. has organic production standards, as does most of the rest of the world for organic produced products that do not, again, have antibiotics. And back to our Pfizer bulletin, the criteria are pretty much the same. I think that we have to consider that it needs to be easy, economical, and consistent. In other words, it needs to be affordable to be used on a mass quantity. It needs to be easy to administer, worked in a feed mill, etc. And you have to have a consistent response. In other words, every time you put it into that feed, those animals need to respond the same way, in the same fashion every time. And then, of course, as we've been talking about this week, the other factor then is gut health, pathogen reduction, antibiotic resistance reduction. And this one is more of a new thing in the idea that some products are now able to reduce antibiotic resistance within that gut microbiome, and that becomes a part of the factor as well. And again, we'll talk a little bit about some of the mechanisms on that. But we're going to start with, as far as criteria, what I'm going to talk about today is mostly on the pathogen reduction side. And part of that's reason because of all the different criteria, it's probably the easiest one to assess on the front end in terms of making decisions, in terms of where you want to go with a feed additive and how you want to screen those feed additives. So let's talk a little bit about alternatives to antibiotics. And I put them into two broad categories, and there's others obviously that we've talked about this week that would fit under one or the other of these categories. The first group I would call would be inhibitory to pathogens. These might be ones that you would administer to clear out a pathogen that has already been colonized. And so we go from what I would call fairly broad spectrum, which would be essential oils and related compounds, fairly nonspecific, could affect a multitude of different bacterial species within a gut microbiome or within other microbiomes. Organic acids are also probably somewhat nonspecific. You would expect anaerobes, for example, that produce organic acids to be more resistant than, say, a facultative organism or whatever. Bacteriocins, which are going to be somewhat more specific and probably target only certain groups of organisms potentially, such as gram-positives. And then finally, bacteriophages, which would be highly specific. A good example is salmonella and bacteriophage. I mean, you have 2,600 serovars. You have phages that are specific for particular serovars, and in some cases maybe even just for specific strains. And so you have a multitude of different specificities in terms of your compounds that are inhibitory. Then we have another set that I refer to as preventative. We have vaccines, which stimulate hosts' immune response. You work with an attenuated strain of a particular pathogen, you inject that, or you administer it orally, and then the host responds to that immune response. The idea is then, of course, when the wild-type salmonella, for example, comes in, then the host is able to mount a specific immune response to that. Probiotics, which we've seen a lot of discussion on that this week, live, good bacteria that are added externally to the gut with the hope or plan that they establish in the gut and help to accentuate a quote-unquote healthy type microbiome. Prebiotics, which I'm actually going to spend quite a bit of time on because that's what we work with mostly. Feed additives that select for already in place good gut bacteria. Some of these could be things that you would typically find in probiotics as well, but generally these bacteria are already established in the gut, and the prebiotics basically select for them. And then finally, the concept, and it's kind of a natural projection, is symbiotics, where you take a prebiotic, you administer that simultaneously with a probiotic, and the idea is a probiotic can use that prebiotic as a substrate, and that helps it get established in the gut. So I'm not going to spend a lot of time on probiotics. You've seen quite a bit of discussion on that in the past, and at this meeting, basically the WHO definition that was laid out a few years ago was basically live organisms which, when administered, confer quote-unquote a health benefit on the host. And I mean, this has been around a long time, over 100 some years, and so the antibiotic era somewhat displaced that, but now we're back into the era again of really looking hard at probiotics again. And they certainly have reemerged. Common probiotic bacteria, Bifidobacterium, Lactobacillus, and of course Bacillus has been talked about quite a bit this week in terms of germination schemes and those sorts of things to get Bacillus established. A multitude of health benefits, and a lot of these are related to human health benefits. That's a very partial list. I mean, resistance towards enteropathogens, of course, is a driver, combats diarrhea, relief of lactose intolerance, serum cholesterol, improve immune function, which has been talked about quite a bit this week, cancer prevention, but it's a very long list. Some of these are more anecdotal than others. Others are better documented. I mean, you'll see certain reviews will come out and say, one review will say it'll combat diarrhea, and another one will say it'll combat constipation. So you kind of have to sort of kind of pick through this and figure out which ones of these are anecdotal and which ones of these are more mechanistically sound or make sense from a biological standpoint. So let's go to prebiotics. Prebiotics, and you probably are very familiar with this already, but these are basically what we call non-digestible food or feed ingredients. They give detectable host benefits. They're not absorbed or utilized by the host. This was the definition that was originally laid out by Glenn Gibson back in 1995, and we've kind of used or worked off of that as a model ever since then. There's a class of oligosaccharides that falls under that, fructooligosaccharides, galactooligosaccharides, and then mannans or yeast cell wall compounds. All of these can be utilized by what's quote-unquote good bacteria such as Bifidobacterium and Lactobacillus. Again, this concept of probiotic where you could feed one of these to a probiotic along with a probiotic strain, get a symbiotic type effect. That would help the probiotic to get established in the gut, and by basically supplying it a specific substrate that it can use. The question gets to be, and we started to realize is it's not just Lactobacillus and Bifidobacterium that's probably responding to a lot of these prebiotics. There are probably other gut bacteria as well, and that are capable of utilizing these prebiotics and probably do different things with them as well, not just some of the metabolism we understand with our lactics, but actually do some other fermentation products, et cetera. And there probably is an overall gut microbe population response as well, and I'll get into that a little bit later in terms of what we mean by that in terms of overall gut response. So obviously sequencing has dramatically changed how we approach and look at these things, and we've had a lot of talks this week where people have used these types of approaches to understand and get a better idea of, a comprehension of what's going on from a gut microbiome. I mean, I came up in the era where we still did anaerobic culturing to characterize bacteria, and then up into the 70s and into the 80s, that was our primary way that we did it. And if you go back into that literature, it still has utility to this day. I mean, a lot of what we saw in that early literature, we actually are picking up on some of the sequencing that we do now in terms of identification of key species and that sort of thing. And so I think there is some reason to go back to some of these methods on some of this to help document or support or fill in some of the blanks on what we're learning from the microbiome standpoint. Of course, denatured gradient gel electrophoresis based off of 16S was used for quite a while as another way to get at shifts in populations and that sort of thing. But all of this was fairly limited information. I mean, cultural methods probably only captured about 10 to 15%, depending on the estimates and how good your culture techniques were, and how well you captured the strict anaerobes that was in that population. With microbiome sequencing, obviously, we're able to get much better pictures of overall bacterial populations, a much more comprehensive analysis, and then, the key thing here is in-depth taxa identification, both known and unknown. I mean, we can start to distinguish unique genetic sequences and say, yeah, this organism, we may not know what it is, but we do know it can be classified as a species or a genus. So this is some work we did several years ago now where we were looking at pasture flock cecal contents, and this kind of gives you an example of the kind of data that you can generate. We used an Illumina-HiSeq on this, which gave us a little bit more resolution. And the things that came out of this is that we definitely saw a temporal switching in terms of specific microbial groups. Lactobacillus changed out species as we went along, and that's something we've seen on some other work as well. Bacteroides and actinobacteria communities increased as birds became more mature, which is, again, not really a surprise. I mean, some of that we've seen with other methods as well. The key thing that we took out of this, and we looked at FOS and GOS, and then we had plum fibers, kind of a non-prebiotic control, just a little bit more of a neutral type compound to see if there was distinct prebiotic effects. The prebiotics tend to accelerate maturation of the bacterial community and therefore presented a barrier to salmonella. And that's a key point, as I think, we've talked some in these sessions about the importance of age and the development of microbiome. And I think one of the things we have to think about with a lot of these feed additives is how do they impact the development and maturity of that microbiome as it goes along. And you can vary that in birds to some extent. And, and that's why some of the in ovo applications these days are starting to be looked at more closely with some of the prebiotics and probiotics as well, is because the idea is let's get an earlier head start in terms of establishment of a more mature gut and a more mature gut microbial population. The other thing that we picked up on this is, as I said, the barrier to salmonella because we had early development. Contrary to that, Campylobacter liked the fact that we developed an earlier microbiota as well, because Campylobacter actually coincided with these increases in these bacterial specific groups. Normally we see Campylobacter at, say, maybe a couple of weeks. We started to see it earlier with some of these prebiotics and some of these where we saw a faster rate of maturity in terms of the microbiota, and there we also saw increases in Campylobacter at an early age. So it's a little bit of a, so depending on the pathogen, it's a little bit of a challenge sometimes to get one size that fits all. And one of my take-home messages early on is I'd say I don't think it's possible. I think we have to really take this pathogen by case to some extent in terms of how we strategize and apply these sorts of types of feed additives. So a different study along these lines is we looked at some yeast prebiotics in commercial flocks, and here we compared that with antibiotic. The antibiotic we looked at on these studies was bacitracin, which was commonly used in some of these commercial flocks at the time. And this gets back to my maturity point a little bit that I was emphasizing in the last slide, is Again, here, what we noticed with the antibiotic feeding was that Campylobacter significantly increased in the antibiotic-fed birds by 4 weeks versus both the control and the prebiotic. The prebiotic and the control pretty much behave the same way. Lactobacillus, on the other hand, significantly was reduced by the antibiotic at 2 weeks versus control and prebiotic. Now, note that everything really kind of stabilized by 4 weeks. I mean, nothing really was much different by then, or by 6 weeks, I should say. And which is something we see a lot of, is there's a window of development and change in the microbiome, but once you hit 42 days or towards the end of that broiler, things really start to kind of even out, and so your populations oftentimes at the end are really not necessarily that different regardless of the treatment. And one of the things we feel pretty strongly about is you really do need to look at age when you're looking at developing these microbiological populations, because things happen in here that you would never pick up if you just went to 42 days for your samples. So all the treatments were the same by 6 weeks. The question gets to be is there shifts in other groups as well? And what if we change out prebiotics? Is that going to change and different GI tract groups are going to be affected? The other thing I would point out here is that we do see somewhat of an antagonistic relationship between lactobacillus and Campylobacter. We've picked this up on other studies as well, as where how the lactobacillus population behaves somewhat dictates what Campylobacter is going to do, suggesting that there may be a bit of an antagonism between the two. And we've continued to explore that some and kind of want to confirm that and start to understand why that might be to some extent. So let's revisit our prebiotic definition again. And this was a review paper that came out from Hudson's group up in Nebraska 2 or 3 years ago now. And this is a good example of how our resolution and capabilities of doing sequencing and characterizing microbiomes has really changed our thinking in terms of feed additives in general, and certainly specific ones such as prebiotics. Their point was that basically more compounds probably have prebiotic type properties. Okay, it's not just your short list of GOS, FOS, and some of the mannans, but there probably is a much larger grouping of things that you could somewhat consider as prebiotics, or at least have components in them that would behave like prebiotics. And so technically the definition doesn't change. It's still detectable host benefit. And I put the word in detectable because if you can't detect the host benefit, then it starts to be questionable as to whether it truly is a prebiotic or not. So you need to define what that host benefit is going to be, and it needs to be detectable. And so soluble and insoluble fibers potentially qualify as at least being sources of prebiotics, resistant starch, etc. And there's some controversy on this. Some people would disagree with this and be what I would call more purist about it and say, no, these things aren't technically prebiotics, but they do impact the microbiome, and they do elicit some, at least in quotes, prebiotic type properties. So the other thing to keep in mind here is once we start getting into these complex sources of prebiotics, we're looking at what I would call components within that source versus the composite of that source. And we'll go through a little bit of what I mean by that in a minute. But the point is when you get into complex sources such as cereal grain or whatever, how the microbiome responds to that entire entity versus how, if you start fractionating it out, varies, and it's somewhat unpredictable as well. And sometimes it's a requirement of that composite. You need that composite to get a complete microbiome effect. I mean, if you start pulling out individual carbohydrates and that sort of thing, you start to lose some of that effect. So there's some suggestion that there's synergism within the components of a particular source of prebiotics, and that's something that we have to consider as well. What it boils down to is that prebiotics elicit what I call a complex functionality. And I mean, I think that's some of the take-home message from some of the work and some of the reviews that were put out in recent times. So we still see a modulation of the microbial community, both structure and function. In other words, composition changes certainly to some extent, but it doesn't stop there. I think one of the things that we lose sight of with microbial populations and anaerobes in the ceca, whatever we're looking at, is most of them are capable of multiple fermentation pathways. I mean, under some circumstances they might make lactate, other circumstances they make acetate propionate, and there's a whole lot of other potential end products they make, and those change depending on substrate availability, passage rate, other organisms around that might potentially cross-feed, for example. And we have to take all those things into consideration when we're looking at this composite microbial population. So yes, we see selectivity in terms of impact on microbial composition, but as that prebiotic structure or source becomes more complex, we also probably have a much more complex consortia response as well. More organisms are involved, there's more interactions going on, so on and so forth. And so we end up having to classify this response both as direct and indirect. By direct, organism sees prebiotic, organism hydrolyzes prebiotic, organism ferments prebiotic and produces end products. But then we have a whole bunch of other organisms who are there as well who take some of those end products, whether it be, say, something like succinate, for example, and then utilize that succinate to produce propionate. And so this whole concept of cross-feeding is something that we probably need to pay more attention to as we get to know our organisms better that we're looking at on some of these cecal contents. And so, and I trained as a rumen microbiologist, so some of this comes natural to me because in the rumen we see this all the time. There's constant cross-feeding between organisms. I mean, you'll have cellulytics, which primarily hydrolyze cellulose, but then you'll have a whole bunch of organisms that can use those soluble products and basically cross-feed and somewhat interact with the primary polymer degraders. And I think we have to think that way in the ceca as well. I think there's a lot of that going on that we lose sight of or we miss when we're trying to classify and characterize what's going on functionally. So what that means is that, yes, we need to not just do microbiome sequencing and characterization, but we actually need to have what I would call a metabolic systematic screening approaches. We need to not just collect up and learn, find out what the metabolites are, but we need to start putting that in the context of what might be going on here, for example. And we'll go through a little bit of some examples of that. So this is sort of what we've come up with in our lab anyway, when we start to screen or when we start to try to characterize a prebiotic and try to assess whether it's a viable candidate for that we're going to use in a poultry setting. So generally, we start with pathogen reduction. I mean, it's the easiest thing to determine. I mean, does the presence of the prebiotic help to reduce the pathogens in whatever you're looking at? In our particular case, it would be cecal contents. So we start out with in vitro because that allows us to do high throughput where we can screen a lot of compounds fairly rapidly, and that's not an unstandard way to do things. I mean, it allows you to do some dosage determinations, et cetera, and do that fairly rapidly and get an idea and narrow down your focus to the things that you want to then go in and do what we would call the in vivo side of things, where you actually challenge birds with salmonella and the birds are being fed the particular candidate prebiotic that you're interested in. So we generally start there, and following that or simultaneously with that, we also look at the rest of the microbial community because we want to know what the interaction is with the microbial community, with the prebiotic itself, and then can we also characterize that in parallel with what's going on with salmonella? I mean, in other words, there's a lot of cause and effect going on here. Yes, salmonella is reduced, but does that relate to the microbiome that's being selected? And does it relate to the fermentation that the microbiome is doing in the presence of that prebiotic? And so these two things are to some extent interconnected. Now, once we have microbiome analysis, of course, we can go in and determine taxa and determine who's there and are there particular signature organisms, for example, that are responding to that particular prebiotic that you're feeding, et cetera. And then with that, when you want to start comparing different prebiotics and compare with control, then the diversity analysis makes a lot of sense because then you can go in and say, okay, does, birds being fed prebiotic A, do you see a change in the microbial composition over time versus birds that are being fed the prebiotic B, for example? And how does that compare with control? How does that compare with other different types of feed additives, say a probiotic or an organic acid or whatever you want to compare it to? And so both of these kind of go hand in hand. And then finally, obviously, the host gut responses, metabolomics, transcriptomics, and proteomics. You want to know what sort of play does the immune system have in this? And the immune system, of course, interacts with the microbiome and vice versa. And so, again, and some prebiotics can be immunogenic as well. And so this allows you to start factoring some of these things in as well. And it's kind of a three-step process. Basically, you start out with what we call a pathogen screen, and then we get into the gut ecology stuff here. And then finally, the ultimate is bird performance. And something I think we need to keep in mind here is ideally you'd like to have both. You'd like to have pathogen reduction And all of this supports that pathogen reduction. In other words, you have explanations as to why you're seeing pathogen reduction, and you enhance bird performance. In the real world, you're not necessarily going to accomplish both. I mean, you get pathogen reduction, as I said, that's an easy beginning point decision to make. And what you hope for is, at the very least, you don't negatively impact performance. I mean, if you can show that you can maintain bird performance, but now you're getting pathogen reduction, that's still a win. And I think we have to keep that in mind to some extent, is you're not necessarily going to hit a home run and get all of this going at the same time. Ideally, you'd like to, and you certainly want to try. And we'll talk a little bit later about how we need to think about that strategically in terms of how we might want to play this out. So I'm going to go through a series of studies that we did to kind of give you an example of how this works. And don't get, there's a lot of data here, and I'm just going to give you some of the take-home messages out of this and how we process this data and how we use it. And we got into a project a few years ago where we were looking specifically at rice bran. And rice bran is a good choice for us in Arkansas, because Arkansas is the #1 rice producer in the United States. And so there's vast quantities of rice bran available. It's pretty much a byproduct, not a lot done with it. It's used in pet foods and some of those things. And Arkansas is #2 in broilers, so it was a natural marriage between, rice byproducts, there's excess of it, and we've got a lot of birds. So it makes sense to put those two things together and see, is there some benefits here? Could this be used as a feed ingredient? And we didn't go into this naively as well. There had been a lot of evidence from a collaborator we had out in Colorado State, Elizabeth Ryan, who had shown in mice models, et cetera, that some rice bran was inhibitory to salmonella, at least in mice, and that there were certain other host responses that rice bran was beneficial to. And so as part of the collaboration, it was a natural thing to say, well, let's see if we see some of these things in a non-mammalian system such as birds, and which would have some really practical implications. So that's kind of what got us started on this. And this is our cecal in vitro model. I'm not going to go into a lot of detail. It's pretty straightforward. We use a cecal inoculum in an anaerobic dilution solution. We use a koi anaerobic hood. We have the usual redox indicators, et cetera, so we know that we're maintaining it under anaerobic conditions. And we've used this model for a lot of years as kind of what we call an initial screen. The nice thing is we can run a lot of things at the same time and do a lot of comparisons and use that to do some range finding in terms of different sources of prebiotics, dosages, et cetera. And we can spike that with salmonella and then use that as a way to quickly screen whether certain prebiotics are going to be supportive of decreasing salmonella under cecal type conditions. And it approximates to some extent what you see in the ceca, and close enough for us to at least get an idea of what potentially is going to work in the birds when we go to that second phase. So we inoculate with a cecal microbiome on time 0. We inoculated a marker strain, and most of the time we look at Salmonella typhimurium, but we've also looked at Enteritidis. We're looking at other serovars now as well. Most of them tend to behave pretty much the same way in this system, so that's not been a concern. And then off of that, we can pull off salmonella numbers, we can characterize the microbiome, and then we can also do metabolite responses as well, fermentation patterns, et cetera, to try to put those pieces together in terms of what's going on. And in this particular study, we looked at 3 different cultivar sources of rice bran, jasmine, red walls, and calrose. And most of these are California varieties. Actually, they weren't Arkansas varieties. Later on, we did look at some Arkansas varieties. And when we look at the numbers here, the thing to look at here is that, so we have treatment, feed plus cecal plus the rice bran, and then of course feed plus cecal, cecal only, feed only. And the big take home here is that only one of these cultivars, calrose, was actually fairly inhibitory to salmonella. The others we really didn't see. In fact, we actually saw some increases in here in terms of salmonella. And so right away, it jumped out at us that there is a cultivar specificity to this. And there had been evidence from Ryan's group and others that that would be the case, that there would be some cultivar differences. And so we use that as our beginning point to start to try to characterize, okay, what's going on here? We've got a complex source, potentially of prebiotic type properties. We want to characterize that in terms of, how is that affecting the microbiome, and how is that affecting then this whole cecal ecology as we go forward. The other thing I would point out here is that, and I'll come back to this a couple of times, is the adaptations required. One of the things we do in this system is that and this was trial and error a little bit, is lots of times you start an in vitro system time zero where you inoculate the salmonella and you also inoculate the cecal inoculum at the same time. And generally what we've observed is we see a lot less inhibition when we do that, or a lot less effect. And instead what we started doing, and we did some early studies to compare the effects, is we do what we call an adaptation, where we take it out 24 hours where we just incubate with the prebiotic. Then at 24 hours, then we go in and spike with salmonella. And that seems a little bit artificial, but if you think about it from a practical standpoint, it makes a lot of sense. Because from a bird standpoint, generally those birds are going to be consuming that diet for a while, so their ceca is going to be, their microbiome is going to be adapted to whatever the prebiotic is. And so this sort of simulates more of an adaptation period. And again, we see much more inhibition, as you notice here. I mean, we got down to the point where we couldn't even detect salmonella at that 48-hour period over here. I mean, we got quite a bit of inhibition even at 24 hours. And so that adaptation and giving the chance for the microbiome to interact directly with the prebiotic and start fermenting and producing fermentation products is a pretty critical part of the process to really get a realistic viewpoint of what's going on from an overall cecal ecology standpoint. So this is what the taxa looks like, and again, I'm not going to walk through all of this. The take-home messages are pretty much this, and this was done 2 or 3 years ago with CHIME 1.9. I mean, we do CHIME 2, the 2 levels now, so we've gotten away from this. We used a variable for region, hypervariable region. Every variable region has a little bit of advantages and disadvantages in terms of how you want to screen things. The V4 region generally worked pretty well for us for these types of populations and microbiome characterizations. And we characterize from phyla to genera, and we use green genes for our 16S gene database on this to identify actual organisms. Some take-homes here is at 28-day cecal inocula. The major genera was ceca only. We was mostly seeing Enterobacter. When we added in feed, we'd start to see Ruminococcus and Osteospira come in. Makes sense. Ruminococcus is more of a fibrous carbohydrate type degrader, whereas Osteospira is more of a starch degrader, and so it makes some sense that you probably start to see those become prominent. When Calrose was added in on top of this, we also saw a big climb in fecal bacterium as well. Go out to 42 days, again, pretty much the same trend for the ceca only. Now we're seeing some Enterobacter as well as Ruminococcus with feed, Calrose, Osteospira, and Lachnospira. And Lachnospira is an organism, or a group of organisms we've seen off and on quite a bit, and we're trying to track that down a little bit in terms of what Lachnospira can do and why that might make some sense. Calrose, we saw nearly 100-fold decrease in Enterobacter, which lined up with our salmonella data that I'd reported earlier. Now, a point I want to make here is that we've published a couple papers on this. Is one of the things that you have to do when you standardize these cecal and in vitro methods is you really have to be careful of what age of birds you take the inocula from, because as this data points out here, we saw differences between 28-day birds and 42-day birds in terms of source of inoculum. And that, I mean, it's kind of a common sense thing. That makes sense because this is probably a more mature cecal microbiome here than it will be at 28 days, and we've seen that with live birds. But I would also say that both are important because we get back to this whole maturity change in microbiota and that being an important component to consider when we're evaluating feed additives. We do need this information here because we need to know what's going on in those intermediate periods as that microbiome develops and how that responds versus what we see at the end point at 42 days. And frankly, we've also looked at 7-day inoculum sources as well, and we're still working out. I'd actually like to look at even earlier birds as sources of inoculum as well, because I think each different age is going to give you different responses in terms of what you see, and I do think that translates some to overall cecal development as things go along. So this is what the diversity looks like, and again, principal coordinate plots, Venn diagrams to show you what the shared microbiome is between the different days and the incubation times. Alpha and beta diversity. 42 days, no surprise, we're more diverse than 28 days. That fits with what we've seen with live birds as well and in vivo studies. Beta diversity, we saw some separation by treatment, but no difference in incubation time, which was a good thing to know because one of the things you worry about with in vitro incubations, you worry about, okay, is the incubation conditions going to, are you going to lose organisms over time if things aren't anaerobic as you'd like them to be or whatever? And so this indicated that our incubation time was not going to be problem in terms of being able to accurately reflect what we're seeing in terms of microbiota. Venn diagrams, we saw 60% share Jenner for all treatments, so actually quite a bit of the same organism. And this gets back to the point I'm going to raise in a bit about functionality and metabolite responses. 22 unique Jenner could be associated with cow rose incubation. We're still kind of sorting that out a little bit and who's there, if that makes sense. 50% of the Jenner were common for all incubation times. So there's a lot of overlap here. And I think that's something that you have to really take in context when you're looking at this type of data is you're not going to get hugely diverse populations. You're going to get a lot of overlap. And, you know, in fact, we were a little surprised we saw that many unique Jenner in this makeup, in this comparison. So let's look at the metabolite data. And we did metabolomics on this with a GC mass spec. We looked at feed plus cecal only, and then versus cow rows added. The incubation time is 0 versus 24 hours. These are only from 42-day birds. And we classified with both KEGG and PubChem databases. So results, we had over 578 metabolites detected. More than half are unknown. In other words, we really couldn't classify them based on the chemical databases that we had. And we were able to identify 211. And this kind of gives you just an idea of most of them that showed what we would call significant increases in terms versus control. Some take-homes here is that nitrogen metabolism was interesting. We saw a pretty good spike in glutamate. Glutamate's an amino acid you typically find in pretty high concentrations in the ceca anyway, so that wasn't a huge surprise. Methionine, I don't really have an explanation of why we saw increases in methionine, but that was interesting that cow rows seemed to support that. Ornithine and allantoic acid are both reflection of uric acid degradation. And so apparently something's going on in here to where we're in some way affecting uric acid degradation cycles that are going on in the ceca and therefore affecting nitrogen metabolism. Beta-glucosidase inhibitors, pentothal and hexatol. Those actually were probably coming from the rice bran. One of the things that we learned as we got into this is, and I should have known this from a plant standpoint, is plants tend to have inhibitors for beta-glucosidase enzymes as they go along, because as they develop cell wall structure, et cetera, they need to start inhibiting those enzymes so that they can start stabilizing those cell walls. And so it made sense that we were picking these up in the metabolite analysis. Fatty acid synthesis, malonic and pathogenic acids were picked up. Some of these reflection of some of the short-chain fatty acid pathways you'd expect, such as butyrate formation, et cetera. And as I said, most of these are all tenfold increase of cow rows versus control. I would like to go back to this point just a little bit here. I think one of the things that we need to keep in mind when we get into these metabolite analyses is there's a lot of contributions coming from the feed and the substrates and those sorts of things that come into these metabolite analyses, and how things like this impact the overall cecal microbiome we need to take into account. I mean, do these things inhibit beta-glucosidase enzymes in the bacteria themselves? Possibly. The other thing that I didn't point out is we also saw quite a bit of phytate in here as well coming out of that, which again is not another surprise. So I think and I'm guilty of this. I mean, I think I focus mostly from a fermentation, microbial metabolism activity, and what I started to realize is that there's a lot of substrates coming in from the feed materials and the other things that are coming in as well that are coming into this metabolite analysis. And I think going forward, what we have to do is we need to start taking a step back and actually doing what I would call some kind of calculation in terms of accounting for the nitrogen and carbon that's coming into the ceca and trying to account for where it's coming from and how that relates to microbial protein synthesis, et cetera. I mean, we used to do that quite a bit in rumen microbiology, and I think it's relevant in the chicken ceca as well. I think we need to start, not just do these vast metabolite arrays, but we need to actually start tying in what's a plant source, what's a microbial source, and how that balances out from a total carbon and nitrogen flow. And I think that's a direction that we need to consider going in the future. So overall conclusions on our rice studies, certainly potential prebiotic source, but very cultivar specific. We're screening other cultivars now, and we're still continuing to see this. Some cultivars are very effective, others are not. In other gut ecosystems, we see other effects. We've had some intriguing preliminary data that indicate that we see methane inhibition in some of the rumen incubations we've done, and that's something we certainly want to follow up on as well, because that's a whole different type of metabolism, and that was a surprise to us to some extent. It appears that, at least in vitro, we certainly support unique cecal bacteria. We inhibit salmonella, it causes shifts in metabolite profiles. We're, of course, back to the decision tree or the flow chart that I put together earlier. Where we're at next is actually to go in and do in vivo studies now with the dosage and the cultivars that we've defined as being most effective. We want to examine, and the other thing I would put here is we want to examine the entire GIT tract. I'm guilty of mostly focusing on the ceca. I need, and we're encouraging our folks in our program to do this, is we need to look from crop down. Because, for example, in the crop, we have a heavy dosage of lactobacillus, et cetera. There's some indications that certainly in early young birds, there's a lot of overlap in terms of compartments and microbial development. And I think if we're going to evaluate things and their true effects on the gut, we need to start from the top all the way to the bottom. And we need to look at all the compartments and see how those things are affected and how development is affected over time, and then how that influences eventually cecal composition and makeup. And obviously, and we're getting into this now, as we want to get into transcriptomics and immunological responses, start being able to add a layer of host response on top of what we're seeing from a cecal microbiome standpoint. So let me wrap this up by saying, back to our original question, and that is, what is an ideal feed additive for broilers, and how can we assess this? And I think we have to accept the market demand reality. And what I mean by that is antibiotics are gone. They're not coming back. And so we have to take the lessons we've learned from antibiotics and start to apply them to what we're developing now as our new generation of feed additives. And so we need to have the same concepts as we've had before, as both detectable and consistent improvements in pathogen control and or animal performance. Ideally, we want both if we can get it, but certainly we need to be able to, whatever we get, it needs to be a consistent response. We get the same thing every time. And the producer out there that's growing those broilers are going to demand that, and if we don't have that, if it's variable or it's anecdotal, it's probably not going to market well. Expectations and requirements, it should be not only effective consistently, but it should be easy to handle. Things that we don't talk about is how does it handle in the feed mill? Will it stand up to pelleting? Will it stand up to other heat treatments? Will it stand up if you want to combine, say, an organic acid and a prebiotic? Can you do that and not lose efficacy? With rice bran, one of the things that we're working now with is with an egg engineer, because milling affects how you recover bran from that cereal grain. And how you recover that bran probably dictates what its efficacy is going to be. And so we have to work those factors in when we start to consider these things. And so it's, all it does is make it even more complex, but the ultimate goal is to have something that's very consistent over time. I'm a big proponent of this, and I always have been, is we've got to know what's going on. We have to elucidate those mechanisms. We've got to know, okay, if we add this in, who's going to respond, how are they going to ferment it, and what's going to be the outcome of that, and then how does the host respond to that. We have to connect the dots between those all those different points. So we need to know how they work. But I would say just as importantly, we need to know when and why they don't work. Because when we're out there making management recommendations and things and it doesn't work, is it a management problem or is it a problem with the product? And so knowing mechanisms allows a person to predict, well, yeah, if you do this, it's not going to work because of this. And I think that's an important point that we have to make. And finally, I think, I'm a little bit biased on this, perhaps being an academic, but I think peer-reviewed science is a critical part of it. I think we have to have that open availability of the data, all the way to the raw data. I mean, the nice thing with microbiome data now is there's a lot more journals demanding that you deposit the data and it's open access, et cetera, and I think that's all for the good. We need that. We have to have that to make progress. So I think the future in terms of expanding criteria for ideal feed additives is, and I'm guilty of this as well, we've got to get beyond just pathogen reduction and prevention. We need to start focusing on some of the other things that have been talked about in the conference, gut health, immune status, inflammation, how are those things impacted? From a system standpoint, performance, welfare, feed conversion, economics, all that needs to be factored in. I mean, there's ways to calculate, okay, if this product costs this much, how much economic impact are we going to get out of that? I mean, does the cost-benefit ratio make sense? And then I think one of the things I've started to realize, I think, is we've got to take a multiple hurdle approach to meet what I call multiple criteria. Back to this pathogen versus performance thing is, probably what we need to start thinking about is that, okay, maybe we need to do combinations that kind of help us accomplish both. Maybe one particular feed additive does help you with pathogen reduction, but then you add in a second feed additive that maybe helps on the performance side, and perhaps there's synergism between the two. Likewise, you know, back to our earlier point about feed additives being inhibitory versus preventative. Combinations of both may make sense. In other words, you put an inhibitory in, such as a phage, to knock out a pathogen population, but then you follow that up with a preventative agent that prevents that pathogen from reestablishing. And I think we have to start having that kind of thinking when we start applying those things. And with that, I will thank you very much for your time, and it's been a complete pleasure being here.
Thank you.